It really is such an honor to be here and to be giving this particular lecture. Because I think that one of the things that I've come to appreciate is many of the things that we're coming to understand molecularly today were seen by others long before us. And in fact, many of the technologies of the past are becoming relevant as we discover new pathways. And so clearly, George Odland and others who have been a part of this department have had major impacts on the field of dermatology. Phil Fleckman, who was mentioned today, was an important mentor to me, colleague and collaborator, who really started the first registry for ichthyosis-related skin types many years ago when the NIH was still invested in funding patient registries. And he was really critical to my career development. And so I'm going to tell you a little bit about my pathway. So I was trained as a classical cell biologist, and yet I found myself working as a cell biologist within a geneticist lab for my MD PhD training, and that was with Rick Lifton, who's now heading up the Rockefeller. And one of the things that Rick really communicated to me was that the biology was important, but that genetics would allow us to make paradigm-shifting discoveries. And so if we think about the utility of genetics in medicine, we really want to understand fundamental mechanisms of disease. We want to be able to achieve early diagnosis, we want to prevent disease, but really we want to be able to identify new targets for therapeutic intervention. And so our population keeps growing. Unfortunately, I've had to update the slide a couple of times. There are 16 billion human alleles on the planet with our population of 8 billion, one from mom, one from dad. And most mutations that are compatible with life are likely present somewhere within the population. In the past, genetic discovery is really driven by intermarriage within families, where we would see rare recessive diseases appearing due to consanguinity. And even the rarest disease in the most remote location comes to attention. And this is primarily happening via instant messaging on apps like LinkedIn or Twitter or something else, where people will be sending me photos of things. And, my kids are always sitting in the back seat like, oh my God, that's gross. But they're coming to appreciate the severity and spectrum of skin disease. And one of the things that people constantly say is, the era of genetics is over. There's nothing left to discover. And what I would say is, we have made incredible strides in understanding the human genome, many of them made here at UW. But despite the fact that we now know that there's 30,000 genes in the genome, we only understand function for about 5,000 of these. And so there's clearly a lot more functionalization of the genome to be done. And not all of that is going to be driven by discovery. Some of it's going to be driven by what you're doing in your clinical practice and the insights that you have. So if we think about the skin as the most beautiful genetic model system there is, it has some important core functions. It's a physical barrier to injury and infection. It's a critical physiologic barrier to water loss, has roles in thermoregulation and communication. All of us have an incredibly large number of cells within our skin, 85 billion. We need to make 3 billion new ones every day. And every time a cell divides, there's an opportunity for errors to be made. And this is why we see somatic mosaicism and new lesions arising upon our skin, in addition to the onslaught of DNA damage that we encounter. If you think about the skin as being a bricks and mortar model, we can think of bricks being things like cells and structural proteins, the mortar being lipids and secreted proteins. And of course, this needs to sit upon a firm foundation, which is the dermis and adhesion proteins. And a single cell type, the keratinocyte, undergoes a regularly and coordinated, orchestrated pattern of gene expression to allow keratinocytes to have different lives within the skin. In the basal layer, they need to stick. And of course, in the suprabasal layers, they need to prepare themselves to create a barrier and also to successfully shed. Anytime you have a defect in the bricks and mortar, we see canonical skin disease. And so the first disorder, lamellar ichthyosis, is due to mutations in transglutaminase 1, which is a really important brick layer, cross-linking interleukin and loricin to form the cornified envelope. Dystrophic EB is due to mutations in one of those adhesion proteins that's really important for the Velcro that makes up the foundation of the skin. And then in this last case, we see an example of somatic mosaicism where, in this case, HRAS is mutated, and we can think of HRAS as being an orchestrator or an architect. And when you have a mutation in HRAS, the architect goes wild and just keeps throwing more bricks upon the pile. So there's a tremendous amount of opportunity that's still out there in genetic skin disease. And part of it comes from the fact that there are still simple Mendelian disorders that we see in clinical practice that we don't yet know the genetic basis of. But we're also able to go after previously intractable Mendelian inherited diseases. And these are primarily dominant traits of high lethality that are primarily due to new mutations that arise within an affected individual that aren't present in another parent that can be found by sequencing parent offspring trios. It also lets us look at traits of high locus heterogeneity, like ichthyosis, that we're going to talk about today, where we know that mutations in over 50 different genes can converge upon barrier dysfunction and cause phenotypically similar disorders. We also can solve somatic or mosaic traits. We can obtain genetic diagnoses. And so all of you, when you go home today, will see mosaicism in your world. And this most commonly occurs in the plant kingdom, but we also see examples in the animal kingdom. And one of my favorite things about giving this lecture is people always send me pictures afterwards of the mosaics that they encounter. And so some of these come from the plant kingdom. They were given to me by Cliff Wheel. And they demonstrate how a single mutation can lead to a single leaf of an impatiens flower that's differentially pigmented, or 50-50 pigmented apple, but it's really this golden retriever that's a coat color mosaic, and this horse, which is also a coat color mosaic, which tell us something else important about mosaicism. Timing is everything. A late mutation is going to give you a relatively localized lesion that you can see upon the skin, and an earlier mutation is going to be more widespread upon the skin. When we see multiple different areas of the skin that are affected, it also tells us that there's likely an increased risk that this mosaic mutation isn't present just within the skin, but is also potentially present within other organ systems. And so timing determines what cell types are involved, what the patterns are that you see, and what the systemic burden of disease is. And so mosaic disorders are incredibly easy to study because we know that there's really just one genetic difference that's present within that patch of affected skin. So we just need to figure out what the affected cell type is. This is often laid manifest for us by patterning. We need to be able to isolate affected DNA, unaffected DNA, and then use some of the incredible tools that we have at our disposal now in human genetics that include exome sequencing, genome sequencing, transcriptome sequencing, and now even there are mosaic disorders that are due to epigenetic silencing as well that we can examine. And so deleterious SNVs that are unique to single nucleotide variants that are unique to affected tissue are likely going to be causative. And so we were like kids in the candy shop, and we just went after every one of these sort of proliferative lesions that we could find in the skin, and each of them taught us interesting things. And so nevus sebaceous being one of the most common hamartomas present in infancy due to mutations in H, N, and KRAS, the same RAS mutations that you see present in malignancies of multiple different organ systems, but here just giving rise to primarily hamartomatous lesions. I actually frequently tell the students in my lab that you can only trust about half of what you read in the scientific literature. And one of the reasons for this is the work that we did in nevus sebaceous. I was working on this and someone said to me, well, don't you know that it's patched mutations that cause nevus sebaceous? And I went back, and in fact, there was an article that said that this was true. But that was also in an era when we thought that the trichoblastomas that were arising within nevus sebaceous were basal cell carcinomas. And so someone used Occam's razor to make a very compelling genetic argument, which ultimately wasn't true. BRAF V600E mutations cause linear syringocystadenoma papilliferum. Chorokeratotic osteal and eccrine dermal duct nevus is due to mutations in GJB2, and yet we see a fundamentally different histology in PEDN than we do in KID syndrome, which is allelic with PEDN. We've done a lot of work in congenital hemangiomas. These are primarily non-involuting lesions that are GLUT1 negative, unresponsive to steroids and propranolol, identifying mutations in RAS, GNA11, and GNA14. And then finally, we did some really fun work on what we would call the cutaneous skeletal hypophosphatemia syndrome, also due to mutations in HK and NRAS that featured nevus sebaceous and giant congenital melanocytic nevi and epidermal nevi in association with really profound bony changes. It turns out all of these individuals had sky-high levels of FGF23 and widespread mosaicism for the RAS mutation right around the time that we would ultimately get some bone tissue and prove that It was actually bone that was releasing, that was causing, it was RAS mutant bone that was causing the release of FGF23. Another group would show that RAS is a really important phosphate sensor. And so when you have hypophosphatemia, RAS is activated, leading to secretion of FGF23. And so what's happening here is you're just getting ectopic expression. So we wanted to move from some of these early wins into thinking about how we could leverage understanding of genetic mosaicism to understand disorders that could ultimately be relevant to common disease. And so John Levinson, who was an MD PhD student in the lab, decided to take on nevus comedonicus with the idea that it might tell us something about common acne. And we recruited 3 patients initially from across the country, and each of them taught us something interesting about the disorder. So what we discovered was that all of these patients were born with smooth patches upon their skin that were obvious but didn't have comedones in them. But as they aged, comedones appeared. And after the comedones appeared, frequently concurrent with the onset of puberty, they would develop large inflammatory cysts akin to severe inflammatory acne. And so we realized that nevus comedonicus was really sort of phenocopying classical acne vulgaris, just in a localized and severe way. One of the other things that we looked at is we saw that these were 2 lesions that were arising on the body, but in this lesion, which arose upon the scalp, what we found is that instead of having hair follicles, we were replacing hair follicles with comedones. And this was a really important recognition for us that we would look at a little bit more deeply when we looked at the histology. And so I was reminiscing with Eleanor Knopp today about the fact that when I was an MD PhD student, I still taught medical students about histology with real microscopes and real slides. And microscopy has been one of the things that has remained a true love of mine in dermatology and in our research group, because there's incredible things that you can see if you just look closely. So shown here is the nevus comedonicus that was isolated from the scalp. And what you can see is the adjacent normal hair follicles have completely normal appearing hair. But when you see the ostium of the comedone, what you see is that you have this really dilated follicular ostia, a very deep cyst. And instead of having a normal glassy lining of the hair follicle, instead you have a highly nucleated lining of the follicle. If we look here on the body, we find that we had normal specification of a follicle, but now this follicle is again filled with laminated keratin. We don't see a distinction between the interfollicular epidermis of the skin and the lining of the follicle, which was beginning to give us some insight into what the potential pathology of this disorder might be. And so John did paired whole exome sequencing in 3 cases, and he found that in each of the cases, there were damaging mutations in NEC9. And what he found was that, in fact, they occurred within the kinase domain of NEC9 and within the RCC1 domain of NEC9. And within the RCC1 domain, they were clustered. And so whenever we see mutations within a kinase domain, we begin to think that they might be gain of function mutations. Turns out the RCC1 domain actually shuts off the kinase domain. And so we thought that, in fact, these mutations might be causing a gain of function. So at the time, we knew that NEC9 is a serine threonine kinase. It regulates cell cycle, checkpoint control, controls spindle dynamics and chromosome separation, but that also it's part of a catalytic cascade within the cell, and that downstream effectors of NEC9 are known to regulate inflammasome activity, which might be bringing us a little bit closer to the predisposition to acne. It also undergoes progressive autophosphorylation before catalytic phosphorylation at threonine 210. And right at about the same time as we were discovering these gain of function mutations, a different group was studying lethal skeletal dysplasia with prenatal eyes in a genetically isolated group of Irish travelers. But what was fascinating was, despite having mutations in the same gene, loss of function mutations led to no skin disease. So we always turn to cells when we have a gene for which we don't fully understand the mutation consequence. And so what we did is we overexpressed our wild-type NEC9 in each of our mutants in keratinocytes. We confirmed that we were expressing NEC9, and then we looked for phosphorylation at that catalytic residue. We found that when we overexpressed wild-type NEC9, we got a bit of phosphorylation at it, but that each of the mutants, whether it was within the kinase domain or the RCC1 domain, led to massive autophosphorylation, consistent with this idea of a gain of function. So thinking back about the histology that we had seen, and at this point, we really didn't have much sense of what NEC9 was doing within the cell outside of cell cytokinesis, we decided to look at what was actually happening within these hair follicles. And so shown on the left is a normal hair follicle, which has K15 localized to the bulge, has K6, which marks the companion layer, AE13, which marks the medulla, and lacks expression of keratin 10, which is a marker of the interfollicular epidermis. But when we look within the comedone and the cyst, we find that all cells are keratin 15 positive. They completely lose expression of hair follicle markers and gain expression of keratin 10, which is an interfollicular epidermis marker. And so essentially what we're seeing is that these gain of function NEC9 mutations are affecting follicular fate. So the follicle is properly specifying, but these NEC9 mutations are making it form skin inside of the follicle rather than a hair. And so we now have a really beautiful mouse model of this, and we're using single cell approaches to understand where that fate decision goes wrong, and thinking more critically about how we might be able to modulate NEC9 to address comitogenesis more broadly. But clearly, cilia dysfunction plays a role in pathogenesis of this disorder. And I wanted to turn to a more clinically relevant story. This is some work that was done by a great postdoc in my lab, Lee Yatsmoni, who is now thankfully joining our faculty, along with some students and one of our rock star clinicians. We became interested in linear porokeratosis, and we recognize that when you see linear porokeratosis, it can have multiple different clinical variants. It can be hypertrophic, it can be inflammatory, and this lesion was actually called ILVEN when it first came in, not porokeratosis. Turns out that for the pathologist in the room, when you're looking at an ILVEN, you have to look really hard to see if there's a coronoid lamella. And we've actually been able to find that many ILVENs are in fact linear inflammatory porokeratosis. And then it also has an atrophic variant, and there are other variants that are not shown here. And so we again used paired whole exome sequencing in these linear porokeratosis lesions. We found that this was a case where there was second hit somatic mosaicism. That is, every one of our initial patients had a germline mutation in PMVK or in MVD, but had acquired a second hit somatic mutation in an early embryonic precursor that gave loss of function within the same gene within a specific region of the skin. And so this established the importance of second hit loss of heterozygosity and second hit mosaicism in these somatic disorders. But the first thing that we do whenever we end up finding mutation is we go back to the pathway. And so we wanted to see where did PMVK and MVD fall within the cell. And it turns out that they are within the mevalonate kinase pathway. And the mevalonate synthesis pathway produces cholesterol and farnesyl pyrophosphate. We know that cholesterol is central to skin barrier function, and we also know that upstream of PMVK and MVD is HMG-CoA reductase. And of course, the most commonly used drug nationwide are statins, which are HMG-CoA reductase inhibitors. And yet none of our patients who have DSAP who are on statins ever get better. And the reason for that is HMG-CoA reductase inhibitors are immediately taken up by the liver via first-pass metabolism. And so we thought, taking a page out of a playbook that had been used for child syndrome, that if we were able to apply a topical HMG-CoA reductase inhibitor, lovastatin, and supply the end product, we might actually be able to improve the disease phenotype. And that is, in fact, exactly what we saw. So this profoundly inflammatory ILVEN had remarkable reduction in inflammation after just 5 weeks of therapy, and has remained near normal at this point with continuous therapy. This more hyperkeratotic lesion present on the hand with occlusion of cholesterol lovastatin ointment saw considerable improvement by 12 weeks and continuous improvement over time. And so we had done this work in linear porokeratosis, which is vanishingly rare, but on the coasts, we have a lot of sailors. And one of the most common things that we see in sailors is disseminated superficial actinic porokeratosis, which a Chinese group had shown about a decade before was also due to mutations in PMVK, MVD, and MVK. But with the therapeutic insight that we had from linear porokeratosis, we realized that we'd also be able to treat DSAP. And shown here are the results of applying cholesterol lovastatin after 4 weeks of therapy and after 12 weeks of therapy. And so we would go on to show that you can actually use just monotherapy with HMG-CoA, topical HMG-CoA reductase inhibitors and achieve similar improvements in disease. And so second hit mutations are really critical to the development of somatic disease. Cholesterol lovastatin is an important pathogenesis-based therapy. Right before the pandemic, we were in the process of patenting and commercializing this and had someone that was going to move this forward. No one ultimately did because you literally can mix it in a blender. The problem that we have clinically is not everyone mixes it quite the same. So there's this incredible variability of compounded products. And so if you have a patient that has DSAP that's not responding to cholesterol lovastatin, the first thing to do is to take a biopsy and send it to the pathologist to make sure it's actually DSAP. The second thing to do is make sure that you're getting a gold standard formulation of cholesterol lovastatin. And I think we really need to move away from 5-FU and all of the other ways that we've tried to treat DSAP over the years that really aren't treating the fundamental genetic defect that's driving disease. I'll tell you a story about vascular malformations or venous malformations that brought together a team from surgery and pathology and my lab. We'd been interested in vascular lesions of the skin, and I told you a bit about our work in non-involuting infantile hemangiomas and congenital hemangiomas. But we also recognize that there's a whole host of other drivers of venous malformations that are recognized within the literature. And so we were approached initially with patients who had hepatic hemangiomas, and the liver's pretty far away from the skin. And so when this first came to me, I was a bit skeptical. But as a favor, I started to take a look at these lesions. And I asked to see what hepatic hemangiomas looked like under the microscope. And it turns out that hepatic hemangiomas used to be called cavernous venous malformations of the liver. And in fact, venous malformations of the skin were also called cavernous venous malformations. And it turns out hepatic hemangiomas are frequently found incidentally when people get pan scanned. They can sometimes have significant morbidity. And of course, we know that venous malformations of the skin can be quite disfiguring. And so I show here some of the similarities of histology between the sinusoidal organization of hepatic hemangiomas within the liver and within the skin. What you can appreciate is that they have shared histologic features with dilated vascular spaces, fibrous stromal bands lined by flattened endothelial cells, and irregular interrupted smooth muscle cells. And so, again, we employed exome sequencing to examine both hepatic hemangiomas and cutaneous venous malformations. What we found is that they all had mutations in a gene called GJA4, and they had a recurrent mutation. So other mutations in this gene can cause completely different disorders, but these clustered mutations ultimately were giving rise to about 90% of hepatic hemangiomas and about 60% of cutaneous venous malformations. And so GJA4 encodes connexin 37, the gap junction protein that's highly expressed in lung vasculature and ovary. And I always am wary of showing too many Westerns in a talk that includes a mixture of scientists and clinicians. But it was really important for us to do some of the fundamental work to understand how these mutations were actually leading to venous malformation. And so all the prior evidence we had was about activation of growth pathways. And we started off looking at things that we know we can drug. We looked at AKT, didn't see any activation of phospho-AKT, didn't see any activation of phospho-ERK. And so we were left struggling for a moment and began to think about what non-canonical growth pathways might be present. And so it turns out SGK1 is also a growth pathway. It's upregulated in hepatocellular carcinoma and other types of cancer, has a pro-survival role in endothelial and vascular smooth muscle cells. We looked specifically at different pathways by which SGK1 could be activated. When we went again and expressed our either wild type or mutant connexin 37 in endothelial cells, we found that there was a specific phosphorylation of SGK1 activating it when we expressed the mutant allele. And so this was important to us because ultimately it turns out SGK1 expression is regulated by aldosterone. And so this again gave us the opportunity to think therapeutically because we know that there are aldosterone antagonists, and that is spironolactone. And so we ultimately would show that spironolactone can reverse all of the cellular changes that are associated with mutant connexin 37. And so regardless of when you add spironolactone to culture medium, you can reverse phosphorylation, that's serine 78, and you can take cells that are really sick and unhappy when they're expressing this mutant and turn them into otherwise wild-type looking cells as soon as they see spironolactone. And so this has revealed to us that for these individuals with hepatic hemangiomas and some cutaneous venous malformations, a relatively benign drug can be used to achieve significant improvement. And so, in the past, we might have said, why would we want to biopsy a vascular lesion? I think this is a good reason for us to start thinking about genotyping a lot of our venous malformations. I'll turn to another story that I have brought here, partly for Dr. Simpson, given his interest in acantholytic disorders. In the midst of COVID, when no one would send us patients and new tissue, we had to turn to study something. And so we started to think about what are the mosaic diseases that might be sitting in our pathology archives that no one has really looked at before. And so what came to mind was Grover's disease, because actually Eleanor Knopp, when she was a dermatopathology fellow, had shown that vulvar acantholytic dermatosis and papillary acantholytic dermatosis was due to somatic mutations in ATP2A2. And ultimately, when we looked at the histology of Grover's, it shared features with these somatic disorders. And so this was interesting to us because we know that Grover's isn't always there. It comes and it goes. We know it has it manifests transient pruritic papules with exacerbating factors, including heat, medication, or radiation. But the histology is really shared with Darier's disease. You see acantholysis, dyskeratosis, hyperkeratosis, and inflammation. And what we really needed was an advance in technology for us to be able to detect the mutations within Grover's disease, because others had used simple sequencing approaches in Grover's and not found mutations in ATP2A2. And so our approach is a little bit different for this. We went into the archive, we found 15 accessions of Grover's disease where there was one biopsy of Grover's and one biopsy of anything else, because we assumed that anything that didn't have the histology of Grover's wouldn't have the mutation. We performed paired extremely high-depth panel-based sequencing of genes known to contribute to acantholysis, and 12 out of our 15 samples showed somatic damaging ATP2A2 mutations that were unique to Grover's disease tissue. But what we found that was even more surprising was that in only 3/4 was the mutation absent from the normal sample, but in 1/4 of these patients, there was actually low-level systemic mosaicism for ATP2A2 mutations. So these would otherwise be patients that could have manifested Darier's disease, but had such a low allele fraction that ultimately what we were seeing was something that looked a lot more like Grover's. And of course, every one of these mutations that we found was a UV signature mutation. So we looked across a variety of different histologic phenotypes of Grover's, and I won't belabor the findings in them here. But what we're seeing is recurrent canonical mutations in ATP2A2. This was really interesting to us because this was one of the first common acquired disorders that we always knew should be due to a mutation in ATP2A2, but no one had ever really proven it. And it makes you start to wonder about all of these acquired disorders that we see in the skin. Turns out that the skin can really tolerate an enormously high mutation burden. And Grover's disease is a classical paradigm of gene-environment interaction. So massive UV exposure gives you a ton of UV signature mutations. One of them just needs to be an ATP2A2. That's present and lying in wait until you encounter an environmental exposure that elicits disease. You can do treatments that make the disease go away, but the disease will always come back again in the future because that clone lies resident within the skin. And so I think this is an interesting opportunity for us to think about how acquired somatic mutation, and we know that certainly mutation within the hematopoietic system gives rise to VEXAS syndrome, a new disorder. There are probably others for us to think about. And so, there's always the question of what comes next, right? And so in the world of mosaic disease, there's still a tremendous number of hematomatous disorders that remain unsolved. There's still some vascular tumors that are unsolved. But really where I think the next generation of discovery is going to come from is linear inflammatory disorders. So as we look at hematomatous disorders, and I'm bringing this to all of you because we have a way to get tissue from any clinic anywhere in the world to our laboratory fast enough to grow cells from it and to do advanced genetic analysis. Sometimes it's actually going to be really meaningful for your patients, which is why I would always say if you have a patient who asks why, say that maybe we can find out. So there are about 60% of cases of nevus comedonicus that remain unsolved. There's panfollicular nevi, milia and plaque, venous malformations, and congenital hemangiomas. Many of them already have singleton mutations and really compelling biologic pathways. We just need more samples to be able to bring things across the finish line. But it's inflammatory disorders where I think we're going to have the greatest opportunity. So we know that all the canonical inflammatory diseases of the skin can also appear in linear manifestations. And this includes lichen planus, psoriasis, vitiligo, lupus, dermatomyositis. And the thing that's really remarkable about these mosaic inflammatory disorders is linear stripes tend to appear early in life. They typically appear in individuals who arise within kindreds that are segregating the common disease through multiple generations, which says that this is likely going to be a stripe that confers additional susceptibilities to an otherwise polygenic multifactorial disease. They tend to be more difficult to treat, and they appear in patterns that are specific to the cell type, linear for keratinocytes, block type for melanocytes, and there are other patterns that we see as well. And so we have proven, and we also believe that investigation of mosaic inflammatory disorders is going to tell us a lot about innate immune determinants of inflammatory disease and potentially open for us new therapeutic approaches. And so just tell your patient that there's someone out there who's interested in it. And if they're interested, send me an email. We can consent the family. We can use saliva mailers to collect saliva. If they don't want new biopsies, we can use old biopsies. There's a lot that we can do now with tissue that's already sitting within your DermPath archives. And we can use a lot of technologies to lead to understanding of disease. And so I'll turn from mosaic disorders that are one of my loves to where I started. And actually the work that I initially did with Paul Cavari was the first ex vivo gene therapy for genetic skin disease, lamellar ichthyosis, which is now the basis of Abeona Therapeutics drug for recessive dystrophic EB. And it was the exposures that I had when I was working with Paul Cavari and going back and forth from Mary Williams Clinic up at UCSF, where I met patients with severe genetic skin disease. And I was convinced as a 20-year-old undergraduate that skin disease was disfiguring, skin disease was meaningful, and that ultimately we need to do better science in order to advance our understanding of these diseases. And it was that sort of moment of epiphany that I had a really long time ago that led me to think more about what we could do in understanding all of the genetic causes of disorders of keratinization. A really large team of people has come together to work on this, including Phil Fleckman and Lynn Millstone. And so the first thing we did is we set out to create a registry. And we had something to work from because Phil had started the registry for ichthyosis a number of years ago. And despite the fact that it had shut down by the time that I came onto the scene, it actually gave us really important ideas about how we might go about obtaining information from patients. And so we were a leg up on phenotyping. So we would get history of photography. We'd obtain blood or saliva from DNA. And ultimately, we would do pre-screening for mutations in what was initially a 20-gene panel, and it just keeps growing as we find new genes for ichthyosis. And those individuals who don't have mutations in known genes undergo exome, genome, transcriptome, and other types of sequencing. And one of the things that's been really remarkable is as we've done this, we've acquired an incredibly large number of patients. Now, about 1,600. We've been able to provide genetic diagnoses for free back to patients, about 1,300 of them. And this actually started with me and a friend of mine who was a nurse working in a coat closet at the patient support meetings for the Foundation for Ichthyosis-Related Skin Types. It was really the investment of a charitable foundation, Doris Duke, that allowed us to get this off the ground and to turn it into a real engine of discovery. And so now we run very formalized pop-up clinics. And this is the group that came with us to our last meeting, where we do comprehensive photography and really fully understand the diseases that we're enrolling within the registry. So if we look at this cohort of patients and we look at the mutations that they have, we find that the great majority of them have mutations in genes that we would all know are likely causes of ichthyosis. About 13% have mutations in 36 other genes that are more rare causes of ichthyosis. But the thing that I really love to do is sit down with our binders and look at the photographs of all of the patients that don't have mutations in known genes. Because what I'll tell you is none of them have read the textbook. Each and every one of them has a disorder that's like nothing you've ever seen before. And in fact, Dee Dee has sent me some photos of a great patient that she has that also hasn't really read the textbook. And so one of the things that I think we face as dermatologists in clinical practice is every once in a while you send out genetic testing and you get an answer back and they say, this is the answer. And then you look at the answer and you say, but this mutation can't possibly cause this disease. And so we need to constantly have our attention driven towards thinking about whether there might be something new. And so I'll tell you about a couple of the new disorders that we found. So dominant lamellar ichthyosis was a unicorn in the literature. It was reported in the 80s by Heiko Trout in 2 families and really not seen again until he sent his samples to us along with Amy Paller and many others. We ultimately found that there were 5 different kindreds that were segregating a dominant form of lamellar ichthyosis through multiple generations. Shown here is the original German kindred, and shown here are 4 others. And one of God's gifts to geneticists are de novo mutations, that is, mutations that arise uniquely in affected individuals and they're not present in parents. What we found is that, in fact, 3 of these kindreds had de novo mutations that were ultimately transmitted, providing really strong evidence that heterozygous ASPRV1 mutations lead to autosomal dominant lamellar ichthyosis. And so we went back and we looked at the phenotype, and we actually often do this backwards. We start by finding the mutation, and then we go back and look at the phenotype. When we did that, we found that there were consistent findings across the different kindreds. And so they had plate-like thick lamellar scale, they had palmoplantar keratoderma, but there was also a spectrum of phenotype that we identified, ranging from patients that had more of a severe ichthyosis vulgaris appearance to patients who had more classical lamellar appearances. Histology was relatively nonspecific, although we saw a degree of hypergranulosis in addition to compact hyperkeratosis. And so, again, we went back to the literature. We wanted to know what ASPRV1 did. It turned out that there was some work that was done in it. It's a single exon gene. It's exclusive to mammals. And it's a 343 amino acid protein that is initially produced in a proform that is ultimately auto-cleaved to a 14 kilodalton active enzyme, and the catalytic residue is known to be asparagine 212. It also homodimerizes, and this is really important because when you have a dominant disease, you need to have a way to make it be a dominant negative, and so it's essentially a toxic cargo to the wild-type allele when it homodimerizes. We also knew that ASPRV1 is thought to cleave filaggrin. So filaggrin is also produced in a proform, which is processed stepwise to form monomers. And these monomers are important for binding the keratin intermediate filaments and making a good cornified envelope. So again, some Westerns. We wanted to look first at what happened when we expressed wild type in each of the mutant ASPRV1 and keratinocytes, specifically with respect to production of cleaved products. And what we found is that each of the mutants that we found had an abnormal band that showed up, which told us that there was a derangement of autocatalytic specificity. But more important was the question of what was happening to filaggrin. So when you do Western analysis of keratinocytes that are exposed to calcium and forced to differentiate, you almost never find high molecular weight filaggrin because it's immediately processed away. And so when we overexpressed wild type ASPRV1, we saw exactly that. But anytime we expressed any of our mutants, we had a tremendous amount of high molecular weight filaggrin. So ultimately, the mutant was blocking filaggrin processing. And so it's the accumulation of high molecular weight filaggrin that's contributing to this disease phenotype. And so this was a remarkable finding for us. And one of the things that's interesting about ASPRV1 is we find that treating these individuals with urea-containing moisturizers can achieve a partial reversion phenotype. And I'll tell one last story, because I think this is another story that highlights for those in clinical practice the real importance of genetic testing. And so Rob Sidbury actually sent me this case here as an individual that he thought had CIE. Angela Hernandez Martin sent me this case from Madrid. This was what we would come to recognize was a novel erythrokeratoderma phenotype. All of these individuals had congenital erythrokeratoderma, or thickening of the skin. They had defective dental enamel. They had abnormal nails. They had absent eyebrows, absent eyelashes, which we didn't initially appreciate, but after we discovered more about the disorder, we were able to learn was hallmark of the disease. They had coarse or curly or absent hair, and they had essentially non-specific histologic findings. And so we had performed exome sequencing in each of these individuals, but I also am very involved with the Foundation for Ichthyosis-related skin types. And I would log into the teleichthyosis website, and every time I'd log into the teleichthyosis website, I'd see a picture of this boy here. And teleichthyosis was sponsored by the Lennox Foundation in memory of Dane Christian Phelps, a little boy who had died unexpectedly with ichthyosis. And when I logged into the website that day, I realized he had no eyebrows or eyelashes. He had an erythrokeratoderma, and he had nail abnormalities. I had met the Phelps family. They came to all of the meetings for first, despite the fact that their child had died. And so my first inclination was to reach out to them and to ask them more. And they told me that he was actually born completely normal and had normal skin for the first 5 months of his life. But at month 5, he started to develop recurrent pustular flares and bacterial infections. He had failure to thrive, but was otherwise doing fairly well until he became acutely ill and was found to have rapid onset heart failure. And despite all of the interventions that could be done by the team at Stanford, he died of his heart failure. One of the things that the family had struggled with was whether to ever do a biopsy on their son. And I asked if there was any tissue that was there, and actually Mary Williams and another one of her pediatric dermatology colleagues had actually biopsied. And I asked the family, have you ever wondered what happened with Dane? Do you think that we might be able to get that biopsy and do some genetics? And in fact, the discovery that we made in Dane would lead us to discover undetected dilated cardiomyopathy in our other index cases, one of whom would go on to transplant. And so ultimately, this would define what we call the erythrokeratoderma cardiomyopathy syndrome. This features congenital erythroderma, defective dental enamel, abnormal nails, and progressive cardiomyopathy. And what's really fascinating is that desmoplakin mutations, which cause EKC syndrome, also cause a whole host of other disorders that range from limited heart disease to acantholytic disease, bullous dermatosis, to classic Caraballo with PPK and woolly hair. But in fact, none of them actually feature generalized erythrokeratoderma. So it turns out that it's specifically these proline substitutions in spectrin repeat 6 that are canonical helix breakers that are driving the unique phenotype that we see in EKC syndrome. So we turned to electron microscopy to see what was happening to the desmosomes in this disorder, given mutations in desmoplakin. What we found is that instead of being normally localized to intracellular junctions, they were accumulating within the cell in inclusions. And as they left the intracellular junctions, we saw a widening of intracellular spaces, but we saw no issues with cornea desmosomes. and there were some defects in secretion of lipid that we saw. And so this gave us some insight into what we thought might be allowing for an epicutaneous sensitization that was leading to that transition from normal skin to severely inflamed skin. And right now, there's an MD PhD student in the lab who's developed a really nice mouse model of this disease that's allowing us to think more critically about what are these inflammatory states and why is ichthyosis red in the first place. But I was called in 2019 about an urgent case, and I was told by the Mickey resident that this was someone that was enrolled in my registry, and they were admitted to the hospital with failure to thrive. They had developed rapid pulmonary edema, were intubated, and their echo showed a dilated cardiomyopathy with an EF of 20%, and they were listed for transplant. And I said, could you just snap a quick photo for me and send it to me as a text? And this is the photo that they sent. And you could immediately appreciate that this was a kiddo who had a congenital erythrokeratoderma with absence of eyebrows and eyelashes. And so I said to myself, this is EKC syndrome until proven otherwise. And so we performed rapid exome sequencing. A couple days later, we had a canonical EKC mutation. And it turns out that Amy Powler and Ellen Irvine had treated a series of patients with desmoglein and desmoplakin mutations with an IL-12/23 inhibitor, ustekinumab, and shown that they actually had improvement of their skin phenotype and a reversal of failure to thrive. What we didn't know is what would happen if you treated a patient who was acutely ill with this disorder. And so based on Amy's prior work, I advised initiation of 12/23 inhibition, and you can only imagine what the PICU team said to me when I said, let's immunosuppress this kiddo. They told me to call pharmacy, and I did. And it was an interesting discussion, but ultimately they agreed to start it. So on the 10th, we were able to get it started. 8 days later, she was extubated. And about a month later, she was off the transplant list. And so not only did 12/23 inhibition reverse the skin disease, it also appeared to reverse the cardiomyopathy. And this is one of the other sort of critical questions that we have here. And so, there is some literature out there, and Dr. Simpson may want to opine on this, that IL-17 drives internalization of desmosomes. It may be that inhibition of IL-17 is part of what improves the cardiomyopathy, but it actually has implications for a lot of different types of desmoplakin-mediated cardiomyopathies. So, this taught us a lot. It taught us that, you can't always know what the phenotype is going to be when you look at the mutation at the outset. And so despite the fact that desmoplakin can cause myriad diseases, we found yet another. We see abnormal desmosome processing that clearly have barrier dysfunction, but also significant immune polarization. I started this work as a human geneticist and as a cell biologist, but in the course of doing it, I discovered that I was working on important diseases that were really orphan disorders that needed to be better studied. And I was lucky to have a series of clinical fellows that have been in the lab, and others have come since this group. And they were really the ones who were able to take all of this clinical information we have, link it with genetic information, and teach us new things about biology. And so Britt had a really beautiful paper on the CARD14-associated papulous squamous eruption that ranges from psoriasis to PRP. We put out a paper that describes 300 new mutations in ichthyosis. We also did important work in looking at the impact on mental health in adults and children with ichthyosis, and have been able to develop 2 new scoring tools that are really critical to moving forward new clinical trials in this disease state. And so I think that all of us who are engaged in the treatment of disease and rare disease have an obligation not only to pushing forward the science, but also thinking about the clinical outcomes. And so I am so grateful to all of the people who have contributed to this work, including people who are sitting here in this room today. I'm incredibly grateful to the patients who have literally contributed of themselves to help us to understand new biology, and also to a variety of different funding organizations. So thank you so much for your attention.
Awesome, Keith. Thank you so much. I'm guessing there's going to be a variety of questions, but I'm going to start with one. And I tell me about DSAP. How does that mutation? I mean, you get UV, it hits one little piece of DNA, it hits your target gene. How do you, is that each one of those then is a clone? And similarly, same thing for Grover's, even more so.
Yeah.
Every one of those just grows, that's one clone?
Each and every one of those spots is its own distinct mutation and distinct clone.
Mhm.
And so one of the things that we also need to think about when we look at these disorders is not every mutation is going to have selective advantage. And so only the mutations that have a selective advantage are actually going to manifest. And so you wonder about Grover's, whether it's actually a small clone that gets to be a big clone when you get the right environmental exposure. Whereas things like ichthyosis with confetti, which is another disorder that we've studied that features revertant mosaicism, you clearly have a genetic advantage of wild type cells over affected neighbors.
So interesting. So I mean, but one cell is, I'm talking very technical here, is one cell is very small.
Yeah.
And these, I mean, so these are huge selective advantages, right?
Huge.
Yeah. Huge. Yeah. And each one, that tells us a lot about the proliferative capacity of those mutations too, huh? Yeah, very cool. Great. Dr. Berry.
So, anytime you have a good clinical question, there's some really good sciences behind it as well. And so I would say that we should have some smart student out there start to work on this.
Can you repeat the question, please? It can't be heard by the Zoom participants.
And so,
Thanks for the comment.
So the question was, why is it that we see regional localization of these spots when these are areas that don't frequently have extended UV exposure, particularly in older individuals? The answer to this is there have been a number of groups that have looked at somatic mutation within the skin. And in fact, we start to accumulate a massive amount of somatic mutation in our skin very early in our life. And likely these clones were selected and created early in our life. And there is something unique about sight and sebaceous lobules and sweat and all of these other things that contributes. And your question about why do we see SKs under the breast? Likely there will be some mechanism that we will find that really sort of tells us why there's a selective advantage in those clones in those sites.
Go ahead, Jane.
So the question here was, we use so many treatments for Grover's that actually induce more DNA damage, like ultraviolet light. And so should we be doing this in the first place? And the answer is those mutations were always there and something elicited them. And once the inflammatory state arises, it's really probably the inflammation that's perpetuating the clonal expansion. And that's really where the scientific, sort of work remains to be done. And so by all means, eliminate the inflammation. And there are now reports of dupilumab, JAK inhibitors, just about everything there is out there as treatments for Grover's, although hopefully we'll have better treatments for acantholytic disorders down the road.
And let's see, Peter turned on his camera. Does that mean Peter's going to say a word to us? Let's see. Okay, but you got to unmute, and I think we can make it work if you unmute.
Can you hear me okay?
Yeah, we hear you. Let me put this closer.
I just wanted to show a cool picture that I found that Dan Lance took of my dad back in September of '85 that I don't think you guys have seen. Can you see that okay?
Dan took that. Okay.
Yeah, and he says on the back, George Odland, September '85, Dermatology Clinic, UW, reviewing charts.
There we go. So, Peter, we got the continuity here, Keith. Peter is a Mohs surgeon who also was in the picture there in the background with one of George's kids. So, yeah.
And I spent 3 years in New Haven learning dermatology.
Yeah, that's another link there. It's great. Thanks, Peter, for turning on your camera and saying hi.
Thank you for being there.
Thank you.
Super. Okay.
Please biopsy them all and send them.
None of that conversation is coming through to the Zoom participants.
The question was. The question was, should we be biopsying ILVEN now that we know that a fraction of the cases are due to mutations in this pathway? And the other question was, how often do you actually see a coronary lamella? And the answer is clinically almost never. But histologically, you can find it if you look for it. And so we section through blocks ultimately on ILVEN. And when we find the coronary lamella, we know that they'll respond to cholesterol lovastatin. But cholesterol lovastatin is also such an innocuous treatment that I would use it for all patients with ILVEN, see if they respond, and then go from there. Yeah, and please biopsy every case of anything linear and inflammatory and put it in the mail and send it to me.
Tell us about that, Keith. You said you have a fancy way to do that, but you then said there's all kinds of low-tech ways to get you tissue in a. Maybe your fancy way was to get you living tissue.
Yeah. So I mean, so the challenge with inflammatory disorders is when you do, we classically core them with a needle to be able to get DNA out of them. And you don't always have a homogeneous population of affected tissue. And so really, we use multiple approaches. We now use a 4 biopsy approach within an individual field of anesthesia, 2 affected, 2 normal. The normal actually really important for us to do a lot of work in immunophenotyping these disorders. I think spatial transcriptomics is something that I never thought in my lifetime would ever exist, but is now teaching us, opening whole new fields of biology. So if we want to understand inflammation of lesions like ILVEN, thinking about how we can prepare ourselves to do that is where 4 biopsies, 1 field of anesthesia is the best way to go. We'll send you a mailer that's everything you need, along with an annotated photo if you send it to us. But we'll take whatever it is that you can provide to us, because we know that doing research while trying to practice clinical medicine is one of the hardest things you could possibly do. And most of the time we do it at the edges rather than at the center of our lives. But it's also one of the things that makes life in academic medicine, I think, the most meaningful.
And you have the consent forms for people, right?
And we can consent them over the phone, yeah. And so we can do it all very quickly, or via an online link, depending on how savvy your patients are.
And Keith, at the super practical level, if people want to do that, it's just an email to you with maybe a photo and a tiny bit of background clinical, and that'll get you started, huh?
Absolutely. The whole reason I love coming to give lectures like this is there's always someone out there who has an interesting case that is ready to share it, and that's what leads to new discovery.